Hi, everyone. It's Stephanie with the Patient Story. Thanks so much for joining us. I'm thrilled with our special guest today, Dr. David Miklos. He's the Associate Chief Blood and Marrow Transplantation at Stanford and also the Clinical Director of the Cancer Cell Therapy Program, which focuses on immunotherapy and CAR T. And how do you introduce yourself to people?
I'm a doctor. I
Okay, yeah, that's good.
people who have cancer. And Stephanie, thank you for this opportunity to speak to your patients. You have a tremendous following, and it's really important that we get the information out to the patients and their families.
Yeah, absolutely. I couldn't agree more. I'm really excited to have you. And so, the first thing, Dr. Miklos, I just would love for you in layman's terms. Most people know what CAR T, cell therapy is. There are a lot of exciting developments happening. Could you give us sort of a lay of the land?
Sure, sure. Stephanie, the use of the patient's own lymphocytes, these are the immune cells that provide us our education. These are the things that we're talking about with COVID, as whether or not you have antibodies or whether you have immune responses, and how are we going to vaccinate ourselves. So we understand the power of the immune system. We know that there were no antibiotics until 150 years ago. We know that we've had 10,000 generations of pond scum up to human beings at this point in evolution, right? And all that depended on our immune system, but we haven't been able to harvest the immune system to cure these aggressive cancers until now. So the opportunities to try to vaccinate a cancer have really been disappointing. And so the general approach of the cancer cell therapy or the CAR T program is to hijack the patient's own immune cells to remove the lymphocytes from the body using a very simple procedure, apheresis, just like you see in the Red Cross donation centers when patients are giving platelets. No pain, 3 hours, piece of cake. Take those cells into the laboratory and introduce a new gene. We call that a chimeric antigen receptor, the CAR. And that modification of the lymphocytes becomes permanent in those cells, but it doesn't go into the rest of the body. You're not going to become GMO like the corn in Iowa. We are modifying the lymphocytes, and those supercharged cells, after, again, 1 week of manufacturing, 1 week of safety evaluation in the lab, are brought back to the patient, frozen in liquid nitrogen. Liquid nitrogen is not just for ice cream. And those cells then introduced back to the patient's body through the same catheter that the earlier lymphocytes came out. They expand from 2 million cells to 20 billion cells in 7 days. It's variable depending on which cell therapy we're talking about, but think about that. That's cells dividing every 7 hours. And frequently, after a week of expansion, the CAR modified T cells represent 50% of the white blood cells in the patient's body. And these are cells that can do four things that have never been done in cancer. These are cells that are proliferating in the patient's body, localizing to the tumor, able to kill with cytotoxic killing like we use for viruses, influenza, and then they persist, providing you surveillance until the cancer's all gone. And in fact, some of these CARs are designed to last for years. Others are designed to simply go away over the next 4 to 12 weeks. So that's the value of the excitement of the CAR T program. It's a paradigm shift of using the patient's own lymphocytes to kill cancer with all the power of the immune system in a way that was never done before. And again, this was pioneered by Dr. Carl June and Steven Rosenberg at University of Pennsylvania and the National Cancer Institute, but really became available broadly to the lymphoma patients only in 2016. And so it's hard for me to remember before February of 2016 when we treated our first patient here at Stanford, since now we are treating 120 patients this year at Stanford with the CAR T cells, and we anticipate increasing about 50% per year. I want to stop there.
That's a great overview, actually. Thank you for painting that picture. I know you really boiled that down for us. And I also understand that some exciting new developments have been happening at Stanford. So let's go ahead and spotlight that with the CAR 22.
Sure, sure. The initial work was targeting CD19. It's a series of cell surface proteins, and they have numbers. Why not just give them a number? So the B lymphocytes that are important in lymphoma and in leukemia, and were the first successful use of CAR T. We have three targets: 19, 20, and 22, for what it's worth. Most of your patients have had rituximab. Rituximab being the monoclonal antibody. I'm sure they recognize that bottle that infuses over 3 to 4 hours of clear liquid. That's targeting CD20, but that's an antibody, right? You put the antibody in and it doesn't grow, it doesn't localize, it doesn't do any of those things we just said. It's effective. Don't stop taking your rituximab. Important part of our therapy. But now we're able to go after additional targets. So if a patient was treated with a Axicell or Kymriah, Tisacel, or soon Lysacel from the Juno work that we expect to be FDA approved. Those are three soon to be commercially approved. Axicell in October 2017 and Juno's Lysacel we expect later this month. Three CAR T cells commercially available that the doctors are choosing which one's the best for that patient, but they're all targeting the same protein. They're all using the same binder on the surface of the cell. They're all very similar, actually. And so creativity means let's go after a new target. One of the biggest problems our research has shown that the patients receiving the CAR therapies who have disease progression frequently 2 to 3 months afterwards, that progression happens in half the patients, and half the patients have long persistent remissions that are out at 3 and 4 years now. But the patients who have the progression, why? Why did the CAR fail? What we're finding to be the most common reason is that the lymphoma cells, the cancer cells, stopped making the target. So what was there before as the target that the CAR bound onto and then interacted with and then was able to activate and kill, when the target disappears, the CAR has nothing to bind. And so antigen loss of CD19 can only be overcome by going to a new target. So here at Stanford, we have been fortunate with our programmatic leader, Crystal Mackel, who came from the National Cancer Institute and has brought some of the basic science constructs that are necessary to move into these new targets. And we've had great success recently with a target of CD22. And so similar to the rest of the CARs, it's a chimeric antigen receptor, uses the patient's own lymphocytes, harvested, but manufactured here at Stanford in our own GMP laboratory, the clean room laboratories like you see with Intel inside, the guys in the white gowns dancing around. That clean room mentality so that we can ensure that the virus goes into the lymphocytes, that it's safely matured, and that we collect enough cells after a week of production and we bring them back to the patient. And so the excitement here is that in patients who had progressive disease after Axicell or Tisacel, that we have witnessed 7 out of 9 of these early patients on clinical trial achieving responses, 5 out of 9 in complete remission. And those durable remissions that are in our lead 2-3 patients are now out to a year. They have very low toxicity, low cytokine release problems. That your patients probably know that what happens after CARs, you feel like you have the flu with fevers and aches and sometimes low blood pressure and sometimes cough or low oxygenation, that we can manage that with steroids and a special drug called Toci. And those toxicities are very low. So that's very exciting to us. Now, please don't run out to all your local hospitals asking for CAR22. This is an investigation being done here at Stanford where we are, again, able to help patients who've had progression after Axicell, and patients do come from all over for this. There are other clinical trials that are also targeting and helping patients who have had progression after the commercially available. One of the most exciting areas there is, instead of using the patient's own lymphocytes, why not take cells that haven't gone through 2 or 3 rounds of chemotherapy, haven't existed for 60 or 70 or 80 years in somebody's body? Why not take a 21-year-old super donor lymphocytes from somebody healthy and fit that have been made stealth-like by removing some of the histocompatibility markers, the immune cells that would be rejected? And those stealth cells are also expressing CAR T cells. Some of the companies that are doing this work are Precision Bioscience and Allogene. They have different targets, CD19, CD20. And again, we're using those opportunities to help our patients here at Stanford. And I'm sure that some of your patients can find those locally in centers near them. The
Mhm.
allogeneic approach is very exciting because you don't have to wait to make the cells. So you already have the cells in the liquid nitrogen freezer, and it's kind of one size fits all. And you know how good those cells are because you've seen them function in other patients previously. So there's potentially a lot of advantages to these allogeneic cells. But of course, we have to make sure that they will grow and expand and not get immunologically rejected by the patient. So these are still early clinical trials, but very exciting opportunities for patients who have not benefited completely, have not gotten a durable response from the CAR Ts that are commercially available. That's how I think about the development of CAR T. I think we're going to talk more about going to other diseases besides aggressive large cell lymphoma, though. So I'll let you ask me the question.
No, but that's great. I mean, I wanted to clarify that population for CAR 22, and as you said, you stressed this is still in trial, so it's not like it's available for people quite yet, right?
That's right. These are clinical trials, which patients can come to centers, academic centers, and participate in clinical trials. And other centers are doing very exciting work. Please don't think that we have all the answers at Stanford. Again, there's very exciting work at, as you would imagine, the top 30 large cancer centers in this country, and we're very fortunate to have the support of the patients and their families.
Perfect. Thank you so much. Look, I love that you're humble and there's a little bit of, that caution, but it's exciting to hear any of these developments because these are options that just haven't been there for people.
That's right. In 2016, before we started our CAR therapies and what turns out to be Axicell, or the commercially trade name that you all know now as Yescarta,
Mhm.
the benchmark study of what happened to patients who didn't go on to CAR T with relapsed refractory lymphoma, who have had 3 lines of therapy previously, was dismal. That was an experience we will never want to repeat. The median time to progression, the median time to death was 5 to 6 months, and the 1 year survival was 15%. And now, with the approaches of Yescarta, Tisacel, uh, and I anticipate Lysacel, the 1 year survivals are 85%. And we think that with having additional therapies that we can use in patients who are progressing, we're going for 100%, right? I mean, what's 85? Who wants 85%, right? Let's go for 100%.